Griffith experiment, reported in 1928 by Frederick Griffith, was the first experiment suggesting that bacteria are capable of transferring genetic information through a process known as transformation. Griffith was studying the possibility of creating a vaccine against pneumonia. He used two strains of Streptococcus pneumonia, S strain which was virulent and R strain which was non-virulent. The R-strain bacteria formed colonies that had well-defined edges and a rough appearance. The rough appearance was due to absence of polysaccharide cord produced by the bacteria. The R bacteria were non-virulent, meaning that they did not cause sickness when injected into a mouse. S-strain bacteria formed colonies that were rounded and smooth. The smooth appearance was due to a polysaccharide cord produced by the bacteria. This cord protected the S bacteria from the mouse immune system, making them virulent, capable of causing disease. In the first set of experiment, he injected the virulent S strain into mice. As you know, S strain is virulent and caused pneumonia in mice and dies when infected. In second step of experiment, he injected the R strain into mice. R strain is non-virulent and doesn't cause pneumonia in mice when infected. The mice stayed alive. In the third step, he killed the S strain bacteria by high temperature. You can see heat killed S strain here. Then he injected the heat killed S strain into mice. Heat killed S strain is non-virulent and doesn't cause pneumonia. The mice stayed alive. At the final step, the heat killed S strain mixed with the live R strain injected into mice. The mice developed pneumonia and died. While the bacteria had been killed, the DNA had survived the heating process and was taken up by the R strain bacteria. S strain DNA contains the genes that form the smooth protective polysaccharide capsule. Equipped with this gene, the former R strain bacteria were now protected from the host immune system and could kill the host. It was found that the virulent trait that was responsible for production of the polysaccharide capsule was passed from the heat-killed S strain cells into the live R strain cells, thus converting the R strain bacteria into S strain bacteria, allowing it to become virulent and lethal by evading the host immune response. Today, we know that the transforming principle Griffith observed was the DNA of the S strain bacteria.